In the early 1950s, the scientific quest to unravel the mysteries of life's origins was teetering on the edge of a groundbreaking revelation. The question that haunted biochemists, geologists, and philosophers alike was how life on Earth began with the inanimate chemicals present on the primordial planet. Amidst this backdrop of curiosity and exploration, two scientists, Stanley Miller and Miller-Urey, embarked on a journey that would forever alter our understanding of the origin of life. In 1952, Stanley Miller was a young graduate student under the mentorship of Miller-Urey at the University of Chicago. Urey, a Nobel laureate, was known for his work on isotopes and theories regarding the early Earth's atmosphere. The time was right for a daring experiment, as theories about Earth's primordial conditions were ripe for testing. Miller, inspired by Urey's ideas and the works of his earlier mentors, was eager to translate these theories into an empirical examination. The goal was clear: Miller aimed to simulate the conditions of the early Earth and observe if organic molecules, the precursors to life, could arise from such an environment. The experimental setup was ingeniously simple yet elegant. Miller constructed a closed apparatus that mirrored the hypothesized conditions of the early Earth's atmosphere. It consisted of a small flask filled with water to simulate the ancient oceans and two electrodes to discharge sparks, resembling lightning. Into this primordial soup, Miller introduced a gaseous mixture of methane, ammonia, hydrogen, and water vapor, ingredients thought to compose Earth's early atmosphere. The entire system was circulated continuously, with sparks firing intermittently to ignite the chemical reactions. For a week, the experiment mimicked the violent and electric conditions believed to have been prevalent on a young Earth. The magic began after just a few days. As Miller peered into the concoction, he noticed a change: the once clear solution had turned a murky brown. This was the first hint of something extraordinary. By the end of the experiment, Miller had conducted a series of analyses to determine what had formed in this brew. To his amazement, the mixture contained amino acids, critical building blocks of proteins, and, by extension, life itself. Glycine, alanine, and several other amino acids had seemingly materialized out of nothing more than simple chemical components and energy. The results were groundbreaking. Here, in a laboratory flask, was a plausible scenario demonstrating how life's essential molecules could form under early Earth-like conditions. Published in 1953, the findings provided a tangible foundation for the abiogenesis hypothesis, the idea that life could arise from non-living matter through natural processes. The Miller-Urey experiment catalyzed further research into the origins of life and opened myriad pathways for inquiry. Although the specific conditions under which life originated remain a subject of ongoing research, the experiment stands as a testament to human curiosity and ingenuity. Though modern understandings of Earth's early atmosphere have evolved, suggesting it might have had different compositions, the experiment's fundamental insight remains valid: organic compounds essential to life can indeed form from simpler chemicals through natural processes. In the great tapestry of scientific discovery, the Miller-Urey experiment is a vibrant thread, reminding us of the power of a well-posed question in a meticulously crafted experiment. It serves as a beacon, inspiring future generations to explore the unknown, armed with curiosity and creativity, forever sparking the possibilities of what might be discovered next. Abiogenesis, the study of how life could have originated from non-living matter, stands as one of the most profound and mysterious inquiries in science. In simple terms, it is the idea that life emerged from a prebiotic world, a chemical-rich environment on early Earth. This theory suggests that, under the right conditions, complex organic molecules self-assembled and gave rise to the first life forms. This single step bridged the gap between inert chemistry and the biochemical life processes we observe today. Abiogenesis begins over 4 billion years ago, when Earth was a very different place than it is now. Volcanic activity was intense, the atmosphere lacked oxygen, and oceans teemed with chemicals. The building blocks for life, such as water, carbon dioxide, methane, and ammonia, filled the environment, and energy sources were abundant. Lightning, ultraviolet radiation, and geothermal energy created a chaotic yet catalytically favorable environment for molecular interactions. These conditions were essential for abiogenesis, setting the stage for the complex interactions that led to the origins of life. Producing molecules like amino acids was just the first step. Abiogenesis also requires self-replicating molecules, like RNA, which could encode and transmit information. The RNA world hypothesis, a leading theory in abiogenesis research, proposes that RNA may have been the first self-replicating molecule. Unlike DNA, RNA is capable of both storing genetic information and catalyzing chemical reactions. This unique feature positions RNA as a potential precursor to more complex life forms. Experiments have shown that RNA molecules can sometimes replicate themselves without the help of other enzymes, supporting the idea that an RNA-dominated world could have existed before DNA-based life emerged. One of the greatest challenges in abiogenesis lies in explaining how molecules like RNA could form spontaneously in nature. The synthesis of RNA requires specific sequences of nucleotides, and even a minor variation in sequence can result in a non-functional molecule. Moreover, the conditions needed for RNA formation are highly specific. Despite these challenges, researchers have demonstrated that certain environmental niches, such as hydrothermal vents or shallow, mineral-rich pools, might offer the conditions necessary for RNA and other molecules to form and evolve. Hydrothermal vents, in particular, are a promising focus. These underwater fissures expel mineral-rich, hot water from beneath Earth's crust, creating localized environments that could sustain unique chemical reactions. This environment may have harbored early life or facilitated the formation of life's basic components. Although we have not fully recreated the process of abiogenesis in the lab, significant strides have been made. Researchers have achieved the synthesis of ribozymes, RNA molecules that can catalyze their own assembly, suggesting that primitive self-replicating molecules could indeed have existed in Earth's primordial conditions. The emergence of such molecules marks a crucial step toward life, as it introduces the potential for evolution and complexity. As these molecules replicated and accumulated, they would have formed larger, more complex systems, eventually leading to primitive cells. Abiogenesis remains a topic full of speculation and active research. While no experiment has yet fully recreated the process that led to life, advances in biochemistry and synthetic biology continue to illuminate how life might have begun. Studying abiogenesis is not only about understanding the origins of life on Earth, it also has implications for finding life beyond our planet. If life can indeed arise from non-living matter, then perhaps the conditions for life are not unique to Earth. By studying abiogenesis, we may come closer to answering whether life exists elsewhere in the cosmos. In this way, abiogenesis is more than just a scientific theory, it is a gateway to understanding the very nature of existence. The story of abiogenesis reminds us that life's origins are rooted in the same physical and chemical laws that govern all matter. Life, in this view, is not something fundamentally separate from the universe, but rather an intrinsic part of it, arising naturally from the conditions of a once barren world. Scientists continue to push the boundaries of knowledge to uncover how life, as we know it, first took hold. And while we may not have all the answers yet, the study of abiogenesis provides us with a rich narrative of how, in a world of simple chemicals and energy, life may have found a way to emerge. Within the profound and intricate dance that life orchestrates on Earth, the assembly of complex molecules holds a place of unparalleled importance. These molecules, macromolecules such as proteins, nucleic acids, carbohydrates, and lipids, are the quintessential building blocks that gave rise to the complexity and diversity of life. Understanding their assembly provides insight into the chemistry that underpins biology, revealing the mechanisms by which simple units combine to create the architecture of life itself. The formation of simple organic molecules on prebiotic Earth, a time when the planet was a crucible of chemical processes. The Miller-Urey experiment famously showcased that basic components like amino acids could form under conditions thought to resemble early Earth, with gases like methane, ammonia, and hydrogen being catalyzed by electrical discharges. Amino acids, nucleotides, simple sugars, and fatty acids thus became the starting line. In the primordial oceans, these molecules would combine in new and complex ways, leading to longer chains and rings, an early testament to life's capacity for complexity. With simple molecules at their disposal, Earth's chemistry forged ahead, assembling monomers into polymers. This step marked the emergence of true biomolecules and laid the groundwork for life's intricate architecture. The genesis of proteins from amino acids involved the formation of peptide bonds in long chains, folding into specific three-dimensional shapes. Proteins emerged as versatile players, capable of catalyzing reactions, enzymes, facilitating movement, and forming structural components like fibers. Simultaneously, nucleotides linked to form nucleic acids, such as RNA and later DNA. The sequential arrangement of nucleotides allowed these molecules to store genetic information, a blueprint for life that could be copied and transmitted across generations. Simple sugars polymerized into complex carbohydrates, providing energy storage and structural integrity, as seen in the cell walls of organisms like plants. Fatty acids converged to form lipid membranes, laying the foundation for cells by creating a hydrophobic barrier essential for separating and regulating internal environments. The birth of polymers marked only the beginning. The final act in the assembly of complex molecules involved their organization and catalytic refinement. As these molecules became enclosed within membranous vesicles, the first primitive cells arose, giving rise to protocells capable of rudimentary metabolism. The self-assembly properties of these molecules cannot be understated. Lipids spontaneously formed bilayers, creating membranes. Proteins folded naturally into active forms crucial for catalysis and interaction. Nucleic acids began to demonstrate autocatalytic properties, particularly RNA in its hypothesized primordial role, capable of both storing information and catalyzing reactions, a dual function that is pivotal in hypothesized RNA world scenarios. Proteins began to drive reactions with precision, allowing for metabolic networks capable of combining molecules in a regulated manner, constructing larger structures and breaking them down for energy. As nucleic acids refined their roles, life achieved the capacity to replicate, ensuring that genetic information was passed on with fidelity, guiding the assembly of proteins through processes like transcription and translation. The assembly of complex molecules is life's foundational dance, one that transforms a chaotic milieu into an ordered, self-sustaining system. It is within this molecular ballet that life's potential is fully realized, with the emergence of metabolism, replication, and cellular organization establishing the premise upon which life's diversity is built. Through the manifold roles of proteins, the strategic storage and expression of information by nucleic acids, the energy and structural contributions of carbohydrates, and the encapsulating abilities of lipids, complex life synthesized from simplicity. This molecular symphony continues to play out, driving evolution and adaptation, radically constructing the scaffolds of existence in a universe predisposed toward complexity. As we extend our knowledge of molecular biology, we continue to peel back the layers of this ancient and ongoing story, bringing us closer to understanding how life's infinite possibilities manifest from such fundamental beginnings. In the warm, swirling soup of oceans and volcanic heat, a precursor to life known as protocells began to form. Imagine a world where life as we understand it today did not exist. Instead, there were the building blocks and conditions for something extraordinary to unfold. Protocells were not alive in the way plants, animals, or even bacteria are today. They were simple, membrane-bound structures, like tiny bubbles, composed of lipids and proteins. These membranes were incredibly important, forming a protective barrier that could encapsulate essential molecules, such as nucleic acids and small proteins. Though primitive, the structure of protocells marked a critical step towards the development of true cellular life. Inside these microspheres, a pocket of potential energy and chemical activity started to brew. As protocells bobbed along in the primordial soup, they had the ability to undergo simple metabolic reactions. This allowed them to maintain a form of homeostasis, albeit rudimentary, just enough to sustain some energy processing and replication of their primitive genetic material. The environment around these protocells was dynamic and often harsh. Volcanic activity, lightning storms, and meteor impacts were frequent, creating an ever-changing cocktail of minerals and compounds. This chaotic environment necessitated an ability to adapt. Some protocells developed mechanisms that allowed them to take in genetic mutations and replicate these changes, setting the stage for natural selection, a staggeringly important mechanism for evolutionary progress. Scientists hypothesize that over millions of years through processes like spontaneous self-assembly and molecular evolution, these protocells gradually became more complex. They acquired more efficient metabolic pathways and began to replicate with greater fidelity, possibly influenced by the catalytic activity of early RNA molecules, a concept delineated in the RNA world hypothesis. This hypothesis suggests that RNA, before the advent of DNA and proteins, played a crucial role as both genetic material and a catalyst. In essence, protocells represent a bridge between chemistry and biology, a stepping stone towards life from the non-living elements of our planet. In the deep recesses of time, far beyond the memory of any living organism, life began on a volatile, infant Earth. It was a world forged in cosmic fire and sculpted by ceaseless tumult from geological and atmospheric chaos. This ancient world, marked by roiling oceans and a thick, tempestuous atmosphere, set the stage for an event that would alter the course of the planet irrevocably, the birth of protocells and the dawn of metabolism. The Earth, during its formative eons nearly 4 billion years ago, was a cauldron of chemical activity. Volcanic eruptions, relentless meteoric impacts, and lightning storms generated a myriad of organic compounds in primordial soups across the planet's surface. Within this fertile chemical landscape, the earliest protocells emerged, simple, yet profound structures that marked the transition from chemistry to biology. These protocells were microscopic vesicles, akin to bubbles surrounded by lipid membranes, which spontaneously formed when lipids were exposed to water. Protocells represented a critical threshold in the origin of life, offering a bounded microenvironment capable of housing the delicate balance of chemical processes necessary for metabolism. These cellular predecessors were not truly alive, but they possessed essential characteristics that hinted at life's potential: compartmentalization, simple biochemical reactions, and the capacity for growth and division. Metabolism in its earliest form was simple. The primordial seas were rich with organic molecules, products of abiotic synthesis spurred by the planet's harsh conditions. These molecules, simple sugars, amino acids, and nucleotides, were absorbed by protocells and used as fuel for fledgling metabolic activities. One of the simplest pathways to emerge was fermentation, a process by which these organic molecules were broken down anaerobically to extract energy in a form that could be utilized for cellular tasks. Glycolysis, the core of this process, allowed protocells to break down glucose to produce small amounts of ATP, providing an initial biochemical energy currency. Meanwhile, in regions of underwater hydrothermal vents, a different story of metabolism was beginning to unfold. The unique environments around these vents, rich in minerals and energy gradients, fostered the origin of chemiosmotic processes. Studies such as those by Martin and Russell in On the Origin of Biochemistry at an Alkaline Hydrothermal Vent illustrate how these vents might have provided the necessary conditions for the evolution of life, especially regarding energy capture and storage in anoxic environments. By harnessing chemical gradients, early life forms could generate energy through proton motive forces, setting foundational processes for ATP synthesis even before the complete development of sophisticated electron transport chains. Despite their simplicity, these metabolic strategies granted early protocells the resilience to survive and proliferate in a hostile world. But not all protocells shared the same evolutionary path. Some acquired pigments capable of capturing sunlight, leading to the dawn of photosynthesis and subsequently altering the planet's atmospheric composition. The metabolic versatility granted by photosynthesis opened new ecological niches and played a pivotal role in transforming Earth's atmosphere to one rich in oxygen, setting the stage for oxidative metabolisms. In the depths of Earth's young oceans, basic organic compounds like amino acids and nucleotides, the fundamental building blocks of life, were taking shape. And soon, these simple structures would form the basis of something extraordinary. From these primordial building blocks emerged a molecular player that would change everything: RNA. Imagine a time when Earth was dominated by what scientists call the RNA world, a stage where RNA molecules began to hold a unique place in the prebiotic world. Unlike DNA, which is designed to store genetic information reliably, RNA could do something special. It could fold into intricate shapes, bending and twisting to form three-dimensional structures. These structures allowed RNA to act not just as a storehouse of information but as a catalyst for chemical reactions, performing some of the same jobs that proteins do in modern cells. In this world of organic molecules, RNA was versatile. It could carry and replicate genetic information, and, crucially, it could promote reactions that made life possible. These early RNA molecules could perform only the simplest reactions. Some randomly assembled into loops and strands that could, under the right conditions, copy themselves. As Earth's environment shifted, its atmosphere filled with new molecules and minerals from volcanic eruptions, RNA was presented with new opportunities to adapt. The RNA molecules that could replicate more effectively or stabilize themselves better survived and thrived. Over countless generations, through a process akin to natural selection, RNA molecules became more complex, and a rudimentary form of genetic inheritance emerged. This ability to replicate meant that RNA molecules could now compete for survival in the shifting landscape of early Earth, and those that persisted passed on their adaptive traits. Then, something extraordinary happened. As RNA molecules proliferated and diversified, they began to interact with another set of molecules in the primordial soup: amino acids. These amino acids floated freely in the early oceans, the products of chance chemical reactions powered by the heat and energy of Earth's young surface. But when RNA met amino acids, the two classes of molecules formed a symbiotic relationship that would change the trajectory of life. At first, the association between RNA and amino acids was a matter of proximity. Some RNA molecules bound to specific amino acids purely by chance, aligning themselves in certain ways. Over time, these interactions became more structured, and certain sequences of RNA began to consistently link up with specific amino acids. Through countless iterations, a pattern emerged: groups of 3 RNA bases, or codons, corresponded to particular amino acids. This pattern, which we now call the genetic code, was like a molecular language, a way of translating the simple, linear information of RNA into the complex, three-dimensional structures of proteins. In this nascent code, sequences like AUG would come to represent methionine, and U would stand for phenylalanine. Each trio of RNA bases had an amino acid counterpart. This seemingly simple pairing between bases and amino acids was revolutionary. It allowed RNA to guide the assembly of amino acids into longer chains, which would become the first primitive proteins. Proteins were unlike anything that had come before. Unlike RNA, which could fold only into limited shapes, proteins could twist, coil, and fold into complex forms. These structures made proteins far more versatile catalysts than RNA alone. Proteins could perform specific chemical reactions, bind to other molecules, and even create structural frameworks within primitive cells. With proteins at its disposal, life's capacity for complexity multiplied. The simple protocells of early Earth, once composed primarily of RNA and lipid membranes, now had access to proteins, which expanded their ability to sustain and control biochemical reactions. The emergence of proteins marked a profound shift. These molecules, constructed through the blueprint of the genetic code, became the engines of metabolism, driving the chemical reactions that made life possible. They could act as enzymes, catalyzing specific reactions with speed and precision. They could also form structural components, providing stability to the delicate cells and helping them survive in the harsh environments of ancient Earth. Proteins allowed life to thrive in conditions that would have otherwise been unbearable, enabling protocells to become more stable, resilient, and capable of growth. In time, the system of RNA-based genetic material, protein enzymes, and cellular membranes became more intricate. But there was one more crucial adaptation on the horizon: DNA. While RNA had served as both a genetic material and a catalyst, it was chemically less stable than DNA. Over millions of years, some RNA-based systems developed the ability to synthesize DNA, a molecule that shared the same genetic code but was far more stable. DNA's double helix structure protected its genetic information from degradation, allowing protocells to store genetic information securely over many generations. With DNA taking over as the primary genetic material, RNA transitioned to an intermediary role, bridging the gap between DNA and proteins. The final piece of the puzzle was in place. DNA stored the genetic instructions, RNA acted as the messenger, and proteins executed the instructions. This system of information storage, transmission, and execution created a cycle of genetic continuity that allowed life to persist, adapt, and evolve. Each generation of cells carried forward the genetic code, passing down the proteins and processes that enabled survival. Over billions of years, this molecular system would support the emergence of more complex organisms, setting the stage for everything from single-celled organisms to plants, animals, and eventually humans. The genetic code, proteins, and DNA together form a molecular symphony that has driven evolution and adaptation, leading to the vast diversity of life we see today. Proteins were unlike anything that had come before. Unlike RNA, which could fold only into limited shapes, proteins could twist, coil, and fold into complex forms. These structures made proteins far more versatile catalysts than RNA alone. Proteins could perform specific chemical reactions, bind to other molecules, and even create structural frameworks within primitive cells. With proteins at its disposal, life's capacity for complexity multiplied. The simple protocells of early Earth, once composed primarily of RNA and lipid membranes, now had access to proteins, which expanded their ability to sustain and control biochemical reactions. The emergence of proteins marked a profound shift. These molecules, constructed through the blueprint of the genetic code, became the engines of metabolism, driving the chemical reactions that made life possible. They could act as enzymes, catalyzing specific reactions with speed and precision. They could also form structural components, providing stability to the delicate cells and helping them survive in the harsh environments of ancient Earth. Proteins allowed life to thrive in conditions that would have otherwise been unbearable, enabling protocells to become more stable, resilient, and capable of growth. In the grand narrative of life's origins, the last stages of prokaryotic cell formation represent a profound leap from mere chemical reactions to the intricate world of cellular organization. As we draw our attention to these final phases, we find ourselves amid a transformative journey that redefined what it means to be alive. At this point, prokaryotic cells, with their lipid membranes and self-replicating RNA, had begun to exhibit characteristics that would mark the dawn of biological complexity. These cells were not simply passive vessels of biochemical activity, they became dynamic entities capable of growth, reproduction, and adaptation. They had transcended their origins as simple vesicles in the primordial soup, emerging as the first true living organisms on Earth. The evolution of these prokaryotic cells brought forth a remarkable diversity of forms and functions. Some cells developed the ability to capture sunlight through photosynthesis, harnessing energy from the sun and converting it into chemical energy stored in organic compounds. This innovation not only provided a new energy source for these cells but also had profound implications for the Earth's atmosphere. The byproducts of photosynthesis released oxygen into the environment, gradually transforming the planet's atmosphere and paving the way for the evolution of aerobic organisms. Meanwhile, other prokaryotic cells adapted to thrive in oxygen-poor environments, developing anaerobic metabolic pathways that allowed them to extract energy from organic materials without the need for oxygen. This adaptability illustrated the remarkable versatility of prokaryotic cells and their ability to exploit various ecological niches, from the depths of the ocean to the soil beneath our feet. This diversification not only enhanced their survival prospects but also set the stage for complex interrelationships within ecosystems. As these cells proliferated, they began to exhibit behaviors that hinted at social organization. Some prokaryotic cells formed clusters or chains, showcasing an early form of multicellularity. These cellular arrangements facilitated communication and cooperation among cells, allowing for the sharing of resources and the establishment of cooperative behaviors. Such interactions hinted at the intricate social structures that would later characterize more complex life forms. The presence of a protective cell wall in many prokaryotes further enhanced their survival. This structure not only provided physical support but also acted as a barrier against environmental challenges, enabling cells to withstand harsh conditions. The rigidity and shape defined by the cell wall contributed to the diversity of forms seen in prokaryotic organisms, from the spherical cocci to the rod-shaped bacilli, each adapted to its ecological niche. Within this dynamic world of prokaryotic life, genetic exchange began to take place. Through processes such as transformation, transduction, and conjugation, prokaryotic cells could share genetic material, facilitating rapid adaptations to changing environments. This horizontal gene transfer allowed for the swift spread of beneficial traits, such as antibiotic resistance or new metabolic capabilities, enhancing the resilience of these early organisms. In this context, the story of prokaryotic cell formation is not merely one of individual cells, it is a tapestry of interconnected lives. These cells, as the first living entities, laid the foundation for the complex web of life that would follow. Their evolutionary successes and adaptations fostered the conditions necessary for the emergence of eukaryotic cells and multicellular organisms. In essence, prokaryotic cells became the architects of ecosystems, shaping the environment and influencing the evolutionary trajectory of all life forms. Thus, in the later stages of prokaryotic cell formation, we witness not just the birth of life but the inception of a vast and intricate biosphere. From simple beginnings, these cells evolved into sophisticated organisms capable of harnessing energy, adapting to their surroundings, and forming complex relationships. Their legacy endures in every living organism on our planet, as they represent the fundamental building blocks of life, a testament to the power of evolution and the relentless drive toward complexity and diversity in the natural world. In the later stages, cyanobacteria slowly began to accumulate oxygen on Earth. Life could no longer rely solely on anaerobic pathways. The evolution of aerobic respiration marked a critical evolutionary leap. It offered an unparalleled increase in energy extraction efficiency, vital for supporting more complex biological systems and encouraging the diversification of life. The work of researchers studying the transition to aerobic metabolisms reveals how protocells adapted through evolutionary pressures to develop these intricate pathways, significantly reshaping life on Earth. Favored by natural selection, protocells with more efficient metabolisms outcompete those less adaptable to changing environmental circumstances. This relentless march towards greater efficiency and complexity brought about the eventual rise of prokaryotic cells with sophisticated metabolic networks and organized genetic repositories. The narrative of these early metabolisms, underscored by ongoing research and sources like Opren's work and investigations into prebiotic chemistry, reflects not only a journey through time but an ongoing story interwoven with the fabric of life itself. The conflation of protocells and emerging metabolisms stands as a beacon of life's ingenuity, demonstrating a profound interplay between environmental conditions and biochemical innovation. This saga not only laid the cornerstones for biology but also underscored the resilience and adaptability inherent within living systems, offering us profound insights into life's possibilities on our planet and beyond. In sum, the tale of protocells and nascent metabolisms is an epic of legendary proportions, a testament to the relentless creativity of life's formative processes in the ancient seas, a narrative still echoed in the complex metabolic pathways of today's organisms, and a reminder of the continuing legacy of evolution's quiet power. Understanding this history is crucial, not just for appreciating the past but for shaping the future of life's exploration in the universe.